So just to introduce myself very quickly, I'm a researcher in La Rochelle in France, and I'm working quite extensively with marine birds and Arctic marine birds, and so I set up this pan-Arctic network almost 10 years ago now, named ARCTOX, and so the idea today was kind of present you this network, present you the objectives we have and some of the results and what we can get from it, and also what could be the next steps. So just to start, I'd like to say that, as I said, it's an international network, and I would like to acknowledge all the people who are involved. It's not only me, it's many people which are part of this network. So I want to mention this here, and so very briefly to give a context of the story and of how this network came first. So as I said, it's all about Arctic and subarctic, mostly at the beginning, and which are, we know, facing accelerating environmental modifications, and among these modifications there's an increase of the environmental pollution, which is linked to many things, but mostly because of the long-range transport of contaminants from countries in the northern hemispheres to the Arctic via atmospheric oceanic currents, via rivers, but also linked to the quick melt of the cryosphere in the Arctic, which is releasing contaminants in the environment, and which is also opening new areas for human activities and polluting human activities. And among all the contaminants, there's many of environmental concern, but the one we are interested in is the mercury. We know this is a mass budget published in the AMAP mercury assessment report recently, which shows that there's a lot of mercury transported to the and deposited to the Arctic every year, and some of this mercury enter the food chains as methyl mercury, which is a toxic form of mercury, and will bioaccumulate along the food chain up to top predators like seabirds, which are facing the highest concentration and which are the most vulnerable to the toxic effects of mercury. Because we know that mercury, even at quite low concentrations, can have high toxic effects as a neurotoxin and also as an endocrine disruptor. So it will affect animal behavior, survival, reproduction, physiology, etc. And so it's even in the Arctic, it's even more of concern as recent analyses as part of AMAP again showed that there's still increasing trends of mercury in the biota in some Arctic regions, which you can see here on this map. Dots, orange or red, shows increasing trends of mercury. So we can see that this is still a concern in many regions. And so in that context, as I said, a few years ago, I was thinking about it, and there was clearly a need for pan-Arctic monitoring to evaluate and understand the contamination of the marine species and the marine food webs in general. And of course, there are many international or regional initiatives which are trying to monitor mercury in the environment at large scale. I just give the example of the AMAP for the Arctic, or the global mercury assessment for the global scale, but there's others. And these initiatives are extremely important, but also they try to combine information. Most of them try to combine existing information, and the thing is they use, as you can see on this map, which shows for AMAP the different species monitored as part of the mercury assessment. Some the medias used are different according to the region. The species of interest are different according to the regions, even the family or the group of species. So that makes sometimes very difficult to compare mercury levels that are monitored throughout the Arctic. And we can also see a big black box in Russia. And even for when we focused on one group of species like seabirds, we can see that some studies are focusing on some tissue like eggs or feathers. Some others are comparing tissues between species or tissues within species. So again, just to say that it's sometimes hard to combine and to compare information collected all around the Arctic. And so in that context, there was a need for coordinated pan-Arctic and community studies to track mercury and complement the existing initiatives. And so that's where I set up ARCTOX in 2014 together with, as I said, many colleagues. And today ARCTOX allows the collection of samples, of seabird samples, at more than 70 sites all around the Arctic, which are represented here by these red dots, thanks to collaboration of colleagues from different countries. And so it allows this collection on a total of 26 seabird species. And so seabirds are important not only because they provide information about their contamination, but also because they can be used as bioindicators of the contamination of the entire marine food web. And also because seabirds, if you take different species will use and reflect different habitats. Some are more coastal, some are more oceanic, some are more benthic or epipelagic. So by using the entire seabird community, we can have an overview of the contamination of the entire system. So that was the idea. And on those birds, we collect two kinds of samples, as blood samples and feather samples, which are very complementary tissues, providing complementary stories. The blood reflecting the short-term contamination, so bird contamination at the time when we collect the samples, so that's during the breeding season in the Arctic. And feather samples, and I'll come back to that, which reflect a longer time of contamination and reflects the winter contamination, which is spent outside of the Arctic. And so over the last 9 years, within ARCTOX, we've been collecting a bit more than 5,000 blood samples and about 10,000 feather samples. So we have quite a large collection of samples. And here you have a map where you can see the different families which are sampled according to the regions. And that's just to show that we have a rather homogeneous and balanced sampling all around the Arctic, also in terms of bird family. So we have within ARCTOX, we have several objectives, which I would like to describe and show a bit here. Of course, the first objective is to talk and to study the bird contamination and how birds are contaminated and thus how they are vulnerable to mercury during the breeding season in the Arctic. And so if we look first very quickly at results, mercury results we measure in blood samples, you here have the mercury concentration, so it's total mercury, but I could come back to that. It's a good proxy for methyl mercury, since most of mercury in the blood is as methyl mercury. So you have the mercury concentration for the different study species, and the first thing you can see is a high variability between species, with the most contaminated one, like the great skua, being something like 20 times more contaminated than the least ones, like the auklets in the Pacific. And this graph actually makes sense because it mostly reflects differences in trophic ecology and in diet between the different species, with the piscivorous ones being more contaminated, higher in the food chain than the lower planktivorous ones. But what does that mean? The question is, okay, we have the levels of mercury, but what does that mean in terms of risk for the birds? And so what we can do is we can categorize mercury levels in risk categories. So Ackerman and colleagues published these risk categories a few years ago, from no risk to a severe risk linked to mercury according to the concentrations. And if we look at our different species, and here you have the percentage of individual that fall in each category according to the species. And what we found is basically 50% of the birds we are studying, or we have been collected samples for in ARCTOX, are above the non-adverse toxicity threshold, and we have 1% which are falling in the moderate or high risk categories. So, I mean, low risk is starts to have sublethal physiological effects. I'll come back on it a bit later. But this is, as I said, this is on large species level. But then we can have a look at the population level, and for each species, what we can do. So here you have an example for the brownish guillemot. What we can do for each species is to look at the mercury levels here according to the different sampled population. So here each box plot is a population which is classified according to the longitude from the west, from the Bering Sea to Russia in the east. And what we can see here again within the species at the population level is a high variability between populations, with the highest population being again like more than 10-15 times more contaminated than the least contaminated populations. So high variability, and you can also see some spatial trends with this population in Canada or West Greenland being far more contaminated and above the low toxicity threshold compared to the European population. And again, you can do this graph with the risk categories, and what you see here is again that you see this population, like as I said, Canada, West Greenland, where all the birds fall in the low risk toxicity category. So of course, one could easily say that, okay, birds are in the low category, and so they have some sublethal effect, but that's okay. Actually, it's important to keep in mind that above this 1 microgram per gram concentration, so above this low toxicity threshold, we know that mercury can alter gene expression, hormones, and behavior. But most importantly, we have to keep in mind that this mercury sublethal effect are part of a multi-stressor environment. Birds are not stressed only by mercury, but by many other environmental conditions. And we know that these other stressors could exacerbate the effects of mercury. And just to illustrate this, I give you this example of the little auk. So we are following a population of little auks, which is a tiny seabirds in East Greenland here. And on those birds, we have been deploying accelerometers. So accelerometers are some tiny electronic devices that we tape on the birds, and which will provide information, continuous recordings of the 3D axis and the position of the bird. And from that, we can infer bird behavior, and we can infer its time activity budget, so that the time the bird is being in each activity, and we can infer its diving and foraging behavior, since these birds are diving to collect their prey underwater to 30-40 meters depth. And in parallel, we collected blood samples on those same birds to measure mercury contamination. And what we found here is, on this graph, you have the interval, interdive interval duration according to the length of the dives. And we could see that for the birds which have low mercury contamination in blue, the interdive time is quite similar, whatever the dive duration. But the more the birds are contaminated, the longer are the interdive durations. So an effect of mercury on the interdive duration, which is likely due to a physiological effect on the carrying capacity of oxygen. And why is this interesting? It's interesting because we looked, using still accelerometers, we looked over multiple years at the diving behavior and dive durations according to the environmental condition. And what we found is with a warming of sea surface temperature, birds tend to spend more time diving and having longer dives, which is mostly due to the replacement of the change of prey available to birds and the arrival in the system of prey of lower quality. So birds have to spend more time diving and must have longer dive duration. But that means here that because mercury is affecting the interdive interval time, these sublethal effects of mercury might limit bird capacity to respond to the warming condition and to climate change by increasing again and again the dive duration. So this is to say that although we could think that birds are falling in this low risk category, we should not neglect these potential sublethal effects that could be exacerbated by other environmental stressors. But when we talk about mercury toxicity and the risk of mercury, we also have to keep in mind the role of selenium. Selenium, which is an essential element, but which also has a protective effect against mercury toxicity. So I won't go into physiological details here, but to illustrate this, I'll show you this study in Antarctica, actually, where colleagues have studied two populations of two species of skuas, the South Polar skua and the brown skua, and they measured they estimated the population growth rate according to mercury levels. And what they found is in both cases an effect of mercury on the population growth rate, with a decreased rate with an increase of mercury contamination. But as you can see here, the species with the lowest levels of mercury, the South Polar skuas, also has the higher impact on the population rate, while the brown skuas have higher levels of mercury, but also lower impacts on the growth rate. And this is actually explained by the levels of selenium. What they found is for this population with low levels of mercury, they had even lower levels of selenium. So mercury was in excess in this case, and that could lead to this high decline of growth rate. In the case of brown skuas, yes, they had higher levels of mercury, but they also had even higher levels of selenium, so increasing the protections against mercury toxicity, and so lower effects on the growth rate. So what we did here is within ARCTOX, sorry, I can see that this map is not clear to read, but we used the samples collected within ARCTOX on two species, the brownish guillemot and the kittiwakes, to investigate selenium levels all around the Arctic between these two species. And what we found here, so here, again, sorry for the quality of the figure, but here you have mercury levels in blood according to the longitude, and what we find here is what I've shown before, some spatial trends with higher levels in guillemots in Canada, West Greenland, compared to the European Arctic, and no really trend in the Black Lake kittiwake. But if we looked at selenium levels, what we found is a completely different pattern. First, a big difference between the two species, with selenium levels much higher in kittiwakes than in guillemots, but also some spatial trends with an increase with longitude of selenium levels or in kittiwakes and rather homogeneous level with longitude in guillemots. And if we look at then at the molar ratio, selenium mercury, we see again an increasing trend with longitude in brownish guillemot and no trend in kittiwakes. So what that shows to me here, it's a very preliminary result, but that shows the importance of considering selenium, because selenium varies in space, it varies between species, and it varies differently both spatially and between species. It's different than what we observe in mercury. So it's important to continue studying the effects of mercury on the species to simultaneously track selenium levels, because there might be different responses. And here what that suggests is a higher protection of selenium for kittiwakes on the east side of the Atlantic because of high levels of selenium, low levels of mercury. And for guillemots, higher mercury toxicity risk on the west side, Canada, West Greenland, because of higher levels of mercury but low levels of selenium. The second objective of ARCTOX, now we know a bit more about these mercury levels and the risk associated during the breeding season, is to understand the sources of this mercury contamination. And first of all, the spatial sources, because most of the species that breed in the Arctic, which are in the Arctic for some months, migrate, and they migrate outside of the Arctic, and outside of the Arctic during the non-breeding season. We have very little information about their mercury contamination. So what we do as part of ARCTOX is to use feathers which provide information about bird contamination during specifically this non-breeding period. Just to very briefly explain this to you, we know that mercury accumulates in body tissues over time. Sorry, this is not as a slideshow, so it doesn't appear step by step, but we know that mercury accumulates in body tissues over time. So, but feather molt, at the molt of feathers, it's a mechanism by which mercury that has been accumulated is excreted to the feathers. That means that mercury in the feathers will reflect the levels that have been accumulated since the last molt. And basically, for many of those birds, especially the family of the ARCTOX, here I represent a time window with the breeding season, the non-breeding season, and birds have two different molting periods, molting sequence over the year. One complete molt of all their body feathers here right after the breeding season, and one partial molt of only the head feathers here right after the non-breeding period. So that means that if we go to the field and we collect these two kinds of feather, we'll have information from the head feathers on the mercury that has been accumulated by the birds specifically over the last non-breeding season. And if we collect body feathers, we'll have information of mercury that has been accumulated by the birds specifically since the last molt, so over the breeding season. And so that's actually what we did as part of the PhD project of Céline Albert, that we collected those feathers on 9 different species, ARCTOX species, all around the Arctic, as you can see on this map, in the Pacific, East Atlantic, and West Atlantic. And here's what we found. You here have on this map mercury level measured in the different kinds of feathers, reflecting mercury contamination during the breeding season in red, in green, sorry, and the non-breeding season in red. And so what we found is that birds were about 3 times more contaminated by mercury during the winter time when they were outside of the Arctic, or when they are outside of the Arctic, than during the summer breeding time. And again, you can see here some spatial trends with higher levels of contamination in the West Atlantic compared to the East Atlantic. And so this high and higher contamination during the winter time is very important to consider, because it could have subsequent impacts on the bird, and especially on the bird reproductions. That's what we've seen again with our population of little hawks in East Greenland, where what we did is we tracked the distribution, the non-breeding winter distribution of the birds. So on this map, you have on each point is the distribution of one bird during the winter time. And on those same birds, we collected feathers to know what were their mercury contamination during the same winter. So we could overlap bird position together with their contamination at the same time. And so for all those birds, which were all females, we then followed their reproduction, different reproduction parameters, among which the chick growth. And what we found is a negative relationship between the female head feather mercury levels, reflecting the winter contamination, and their chick growth rate. And so that shows negative carryover effects of the winter distribution and the winter mercury contamination on the subsequent reproduction period and on the chick health. And I'll not go into details, but this is probably due to effects on hormonal effects affecting the parental care. So that was actually first information about the spatial sources of mercury in Arctic and subarctic seabirds. But we wanted to go further and to understand the origin of this contamination during wintertime. And for this, we developed a collaboration together with David Amrou from the lab IPREM in Pau in France to use the stable isotopes of mercury as a tool to investigate the spatial origin of methyl mercury in Arctic seabirds. So very briefly, because I'm not a chemist and I'm not an expert completely, but very briefly, so mercury has 7 different isotopes, and the isotopic fractionation of mercury will provide us information about the biogeochemistry history of the mercury. And there's both a mass-dependent fractionation and a mass-independent fractionation. And the mass-dependent fractionation provides information about, is mostly linked to biotic chemistry, like biotic demethylation, like the reduction in macrobial methylation, the trophic transfer, with some fractionation, and is also due to some abiotic processes like reduction and volatilization. And so the mass-dependent fractionation can be used as a tracer of mercury compounds transformation pathways and how the biotic processes affect the origin of mercury. But there's also a mass-independent fractionation, which is mostly due to photochemistry. And so mercury photodemethylation and photoreduction. But that means that using the isotopes and the mass-independent fractionation in aquatic organisms, we can have an information about the isotopic signature of mercury before it goes into the food web. And so it can be used as a tracer of mercury and methyl mercury sources in the environment, independently of what happens once it's in the food chain. So that's what we did with, again, our little hawks. So what we did is we collected feathers in different little hawk populations all around the North Atlantic Arctic, on which we measured mercury isotopes, and we also measured the carbon isotope, which provides at the same time information about the feeding habitats of the birds. And all those birds were tracked with geolocators, so we could know exactly where were the birds. And so what was the contamination of the birds according to their distribution. So if we look at the results, first of the odd mass-independent fractionation, you here have the isotopic signature according to the latitude and the longitude distribution of the bird. And what we found is a relationship in both cases with a trend in the isotopic signature, spatial trends, which suggests different ocean photochemistry conditions and methyl mercury sources all across the North Atlantic Arctic during winter. And the hypothesis, according to what we know about this signature, was that there could be a higher continental contribution of mercury at lower latitude and more west, and a higher freshwater contribution at higher latitude and towards the east. And so to test this hypothesis, we combine this information together with the stable isotopes of carbon, which I said reflects the habitat and the carbon sources. And what we found again is a relationship that confirms that those levels of isotopic mercury isotopes were linked to a more marine sources and more carbon continental sources here. And if we looked also at the even MIF, which reflects more the atmospheric inputs, again we found a spatial trend with higher wet deposition of mercury at lower latitude and higher dry deposition inputs at higher latitude. So it's I'm not sure I've been completely clear here, but what we find, thanks to the stable isotopes of mercury, is spatial trends of the mass-independent fractionation signature that shows different sources of methyl mercury in our birds during winter, linked to their distribution, with a higher marine and freshwater inputs and higher dry deposition at northeastern latitudes and lower photochemistry, higher continental inputs, and higher wet deposition at south and west latitudes. And finally, the last objective or main objective of ARCTOX is, as I said, not to focus only on seabirds, but to use seabirds as bioindicators of the contamination of the food chain on which they rely, and so use those birds to map the large-scale distribution of mercury in Arctic and subarctic marine ecosystems. And so for that, we have teamed up with a Norwegian program which is named SeaTrack. And SeaTrack aims at tracking birds' distribution at many sites around the Arctic, which are represented here in yellow. So at each of these sites, we have a tracking device that is put on the birds to know about the non-breeding distribution. And at the same time, for each of those tracked birds, we have a collection of feather samples on which we measure mercury and have information about their non-breeding contamination. And so if we, and we have that for several species. So here you have example for 10 species. And here you have the distribution during winter of all these individuals. So one point represents the distribution of one bird during the winter time, and each color is one species. And so on that, we can measure mercury. So we'll have for each bird, each distribution, the mercury concentration, and we can do some spatial analysis to provide these results, which shows the spatial distribution of mercury all around the North Atlantic Arctic, and not only in birds, but basically, I mean, it's a general pattern for the food chain. And so from that, we can first see the longitudinal gradients that we've been mentioning before, with higher concentration towards the west compared to the east. But we can also identify at quite fine scale hotspots and cold spots of mercury that might require specific attention, because these might be spots where species in general and food chains might be in higher risk. And so you can see some of those hotspots here in the southeast Greenland, for instance, or south of the Newfoundland, and cold spot here like around the Faroes, or here southwest Greenland. I just add the variance of the model, which shows that we are actually quite pretty confident on this data and this spatial distribution of mercury that we show. And so the next steps is to try to understand further this distribution. First, because this is a map or figures that has been published recently that shows mercury distribution in zooplankton from a modeling approach, and we could see that even if there's some similarities, there's also quite some differences between what they found in zooplankton and what we found on the food web level, thanks to seabirds. So we have to go a bit further and try to understand those differences. And the next step is also to try to further understand the processes and the drivers of this large-scale distribution of these different hotspots, of these different cold spots. And to that, what we want to do is to combine this map of mercury distribution together with a map of oceanic currents, atmospheric currents, of the human activities, of the atmospheric deposition, to try to further understand why we observe this distribution. So of course there are many things that we would like to do, but the main next steps for ARCTOX are first about temporal changes. We now have 8 years of data and of samples, and we hope to continue this sample collection and analysis over the coming years, and so we could start having an idea of how the levels in the different species, how the levels in the different populations are changing over time, if it's always the same populations that are of higher risk, or if this is changing, and also how the large-scale distribution of mercury and the different hotspots in the North Atlantic, Arctic are changing or not over the time. And the other objective is to go beyond mercury. I think we here have a great network that now could be used to go further and to analyze other contaminants than mercury. And so we are starting to look at it, especially with PFAS. We are doing at the moment some preliminary analysis on PFAS on 2 species at several sites, to also investigate and use this network to see how other contaminants like PFAS are distributed all around the Arctic. So just very quickly to conclude this, I think ARCTOX and the results we have definitely highlight the importance of a coordinated large-scale monitoring of the seabird community to complement the existing monitoring programs and to determine what are the most exposed species and population in the perspective of conservation of these vulnerable species and in the context of environmental change. To understand how Arctic migratory species are exposed to contaminants, not only when they are in the Arctic, but also where they're moving and they go outside of the Arctic. To investigate the impact of contaminant at the large scale, and also to be able to use seabirds as bioindicators to define hotspots of contaminant in the Arctic and subarctic marine food webs, and potentially focus a bit more on these hotspots in terms of species and ecosystem conservation. I think I'm done. Thanks all your attention, and thanks again to collaborators and the funding bodies that are essential for this project.